I'm not going to share my screen yet because I want to talk a little bit about the grading. I have graded Lab 4. So if you did not get 100%, you can resubmit it with the correct answers for 100%, meaning you'll get 5 out of 5. And it must be submitted by 11:59 p.m. this Saturday. I have not graded Lab 5. And if I do get it graded, meaning I will eventually, it will be due next, not this Saturday, but the following Saturday. And I guess I can change that right now. And change the date this one's going to be until, because I haven't got it graded. So it won't be due until November 4th. But obviously I have to get it graded the first time first. All right, any questions about the grading? I don't think I graded anything besides Lab 4 and maybe the lecture worksheet. I will try and get graded labs, actually 00 and 01 for the second time. That is my goal. All right, if there's no questions, let me get out of the grade book and then I'll share my screen. May as well go to Clark. Sorry, I closed the tab that had the grade book, and what I should have done was just move it to the main lecture Canvas page. All right, let me start sharing my screen. And any last questions before we begin? You should see my screen now. Today is October 26th, and we should be starting Chapter 5, Microbial Metabolism. There is a lab today. It'll be Lab 6. It'll be at the normal lab time. There is no quiz this week, so you get a little break there. All right. I'm not hearing any questions, so let's begin with Chapter 5, Microbial Metabolism. The major goals and a brief outline is shown here. Know the terms, what's catabolism, and how catabolism can be broken down into metabolism. Metabolism can be broken down into catabolism and anabolism. Know what activation energy is, what an enzyme is, what the active site of an enzyme is, what competitive inhibition is, non-competitive or allosteric inhibition, what is a redox reaction, what are photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs, what is fermentation, and then what are cyclic and non-cyclic photosynthesis. Any question about any of these terms? All right, also understand what feedback inhibition is. And we're going to talk in great length about aerobic respiration, what it is, its steps, and how it differs from anaerobic respiration. Any questions about what we're covering? So, microbial metabolism is all of the chemical reactions occurring in a microbe. For a non-microbe, it would be all the chemical reactions occurring within an organism. For microbes, because they're all cells, at least all of them except for the worms, which are usually the infective stage is a single cell, the fertilized egg. But for most of the microbes, metabolism is all of the chemical reactions within a cell as well as within the microbe, because the two are the same. for most microbes. We can separate metabolism into catabolism and anabolism. Catabolism is the decomposition reactions, where you have a large complex molecule such as starch, and it's broken down into a simple molecule such as glucose. These reactions are energy releasing, and that's because the bigger molecule, starch, has more energy in it than the simple molecules, glucose. While that reaction occurs, some of the energy in the starch is given off as heat. Heat is just another form of energy. So sometimes you'll hear it, some of the energy is lost as heat. Heat is just another form of energy. So the energy isn't lost, it's just converted to another form. And heat is one form of energy. Some of that energy might be conserved in the molecule of ATP. So some of that energy may be used to combine ADP and inorganic phosphate to make ATP. Now this reaction, or this slide, is a little oversimplified, and it's showing you that catabolic reactions make ATP. That's not the case. In animal cells, there are only two reactions, or two reaction processes, I should say, that make ATP, and that's fermentation and aerobic respiration. In all other catabolic reactions occurring within an animal cell, ATP is not made. And even in a green plant, which has several more ways of making ATP, most of the catabolic reactions do not generate ATP. Any question about any of that? All right. Anabolism is a biosynthesis reaction where you take a simpler molecule, such as glucose, and then you make a larger molecule, such as starch. This requires energy to get this reaction to occur because the glucose has less energy than the starch. So you need to add energy into this reaction. And usually in a cell, for our purposes, we'll just say always, that energy is going to come from ATP. So ATP will be broken down. When it's broken down, energy will be released, and that energy can be used to drive the reaction of adding the glucose together to make the starch. And then the ATP will be broken down into ADP and inorganic phosphate. To my knowledge, all anabolic reactions require an energy source such as ATP. Most of them will use ATP, but occasionally you can have an anabolic reaction which will break down another molecule, usually a high energy phosphate molecule, to drive the reaction. So catabolism releases energy, anabolism gives off energy that is similar to endergonic and exergonic reactions, which I'm not going to talk about in this class, but you may have heard of those in your preliminary biology class, meaning Biology 160 if you took it at Clark. But we're going to define it as catabolism, the breaking down a large molecule into a smaller molecule, and then anabolism, joining small molecules to get a bigger molecule. In anabolic reactions, some of that energy will be given off as heat. So heat, whenever you transfer energy from one form to another, some of that energy is always given off as heat, including in anabolic reactions. Even though energy has to come into this reaction, which is right here, actually, some energy is given off as heat. Any question about any of that? All right. So let's talk a little bit about metabolic pathways. A metabolic pathway is a sequence of enzymatically catalyzed chemical reactions in a cell. You can have, let's blow this up, one enzyme using a starting substrate, molecule A, and making it into molecule B. Molecule B is the product of this reaction. Molecule A is the starting substrate. And then the enzyme, we're just going to call enzyme 1. However, molecule B is the starting substrate for this chemical reaction, which occurs by enzyme 2, converting molecule B to molecule C. So molecule C is the product of this reaction. But then again, molecule C is the starting substrate for enzyme 3, converting molecule C to molecule D. Molecule D can be converted by enzyme 4 to molecule E, and then molecule E can be converted by enzyme 5 to molecule F. So if we put all of these together, we have a metabolic pathway. And B, C, D, and E are intermediate products, meaning they are the product for this simple chemical reaction, but then molecule B is the starting substrate for this chemical reaction. And we call those intermediates or intermediate products. The end product is the last product made, which would be molecule F. And so in the metabolic pathway, the starting substrate will be converted in intermediate steps to molecule F. So molecule F is the end product, the last product made in the enzymatic pathway. Any question about any of that? So metabolic pathways are determined by enzymes. You cannot get them to occur within a cell without the presence of enzymes. And enzymes are encoded by genes. So all metabolic pathways are determined by genes. Any question about any of that? All right. So here are all the chemical reactions occurring within a cell, and we'll talk about each of them, and you have to memorize all of them. Is that clear? Any objections? Only kidding.
Yeah, I object. Thanks.
Let's talk a little bit more about chemical reactions. We will hardly touch on any of these chemical reactions. That might be the Krebs cycle, but then again, it could be there. We will talk about that because we're going to talk about aerobic respiration. But beyond that, let me see if I can blow that up. I can't make any sense out of it even when I blow it up. But that might be the Krebs cycle, in which case we will talk about it. Yeah, that doesn't look like the Krebs cycle there. Oops, didn't mean to do that. Oh, well, let's go there. So let's talk a little bit more about chemical reactions. Chemical reactions occur according to the collision theory. The collision theory states that chemical reactions may occur when atoms, ions, and molecules collide with each other. The activation energy is the energy needed to disrupt the electronic configurations and make the chemical reaction occur. So if two atoms, ions, or molecules collide with each other and have enough energy in the collision, meaning the molecules have enough energy, then the activation energy will be met and the chemical reaction will proceed. The reaction rate is the frequency of collisions with enough energy to bring about the reaction. The reaction rate can be increased by increasing the abundance of enzymes. The more enzymes you have, then they can grab onto more substrates and have the chemical reaction proceed faster. You can also increase it by increasing the temperature. When you increase the temperature, what you're doing is you're speeding up and making the atoms, ions, and molecules move more rapidly. So they have more energy. So if you increase the temperature, you're going to speed up the chemical reaction because each collision will be more likely for a chemical reaction to occur. You can also increase the reaction by increasing the pressure. And this is a little harder to explain, but when you increase the pressure, it's sort of like you're putting the molecules closer together, and then they're more likely to collide. And because they're under more pressure, they also have, when they collide, they're more likely to have more energy, which will increase the chemical reaction. Any question about any of that? All right. Enzymes reduce the activation energy. Oops, trying to get that out of the way just a bit. Of a chemical reaction. So enzymes reduce the activation energy of a chemical reaction. We can have molecule A and B, the starting substrates, also called the reactant, become the product in the chemical reaction. But without the enzyme, you have to elevate their energy that we call the reaction without an enzyme. And if this were, let's say, sucrose, we could convert it into, actually, that's not a good example. If this were glucose, we could convert it into CO2 and water by elevating the temperature, and you take a blowtorch to the glucose, get it on fire, it'll burn, and convert into CO2 and water. Obviously, that's a very high energy level, because that would be raising it to something like 1,000 degrees Fahrenheit. But that's the energy you need for the reaction to proceed without an enzyme. And that we call the activation energy without an enzyme. The increase in the energy level from this part to that part. And then the reaction will proceed. And in this case, glucose will be burnt into CO2 and water. Now, this reaction, glucose burning into CO2 and water, happens all the time in the room that you're currently in. And where is that happening? Come on, somebody's got to state it. Where is that happening in your room? Well, is it happening on your desk? It's happening inside you, in your cells. What's happening is glucose, in the presence of an enzyme, is being converted into CO2 and water. That is nowhere near 1,000 degrees Fahrenheit, now is it? That would kill your cells, cause you to catch on fire too. So the activation energy with an enzyme can be much less than the reaction without an enzyme. And as you know, your cells are only 37 degrees Celsius, which is 98.6 degrees Fahrenheit, which is quite a bit cooler than about 1,000 degrees Fahrenheit. And I don't know what that would be Celsius, but it's very hot. Okay. So that's just showing you that this graph is not, come on, exact on the reaction without an enzyme, because it's quite a bit higher than when an enzyme is present. But the same reaction will occur where glucose will be converted into CO2 and water. And that is the activation energy with an enzyme. Any question about any of that? Come on. All right. Enzymes are biological catalysts. Biological catalysts are simply molecules or molecules or atoms that speed up a chemical reaction. They can speed up a chemical reaction 10 to the 8th times 10 to the 10 times. Enzymes are encoded by genes. A catalyst may not be, like sometimes magnesium or nickel can be a catalyst, but enzymes mostly are biological, actually all of them are biological molecules, most of them being proteins. An enzyme is specific for a single chemical reaction. The specificity of an enzyme is due to the enzyme's three-dimensional shape. Each enzyme acts on a specific substrate. What happens is there will be a specific part of the enzyme which binds to the substrate, and that enzyme will mold specifically to that substrate. So the two combine together. Now, this is a little bit oversimplified. You may have heard of some drugs that can bind to an enzyme and then compete with the substrate for binding to the enzyme. So enzymes occasionally can bind to something other than their one specific substrate. But most of the time, and for this class, we're going to say that an enzyme is specific for one specific substrate. So that the enzyme that binds to sucrose, the enzyme sucrase, will be different for the enzyme lactase, and they don't bind to the same molecules. An enzyme is not used up by a chemical reaction, nor is it altered. So at the start of the chemical reaction, the enzyme will be the same at the end of the chemical reaction. That's not to say that the enzyme isn't changed in the specific reaction. Actually, the enzyme will bind to the substrate and form a complex. This binding of the enzyme to the substrate orients the substrate to increase the probability of a chemical reaction occurring, and that's why it lowers the activation energy. When the enzyme binds to the substrate, it puts stress on the chemical bonds in the substrate to help it form the products, or the product if there's only one. And that lowers the activation energy and increases the probability of a reaction occurring. So that enzyme does need to bind to the substrate and form a complex. When we're talking about enzymes, normally they end in ASE. Some examples: oxyreductase. You don't need to know any of these examples. We'll talk a little bit about ligase, and we will talk about catalase in the lab. Most enzymes end in ACE. This is a convention that was started a long time ago. There are some enzymes that don't end in ACE. To my knowledge, all of them were named before the convention of ending enzymes with ACE began. Like the enzyme renin is an enzyme in the stomach that coagulates milk, and it's very important for babies because it helps them digest milk. And renin was discovered a long, long time ago, long before the convention of ending an enzyme in ACE occurred. Any question about any of that? I should mention that enzymes do not work in a vacuum. Some enzymes require a helper, and that helper can be either a coenzyme. A coenzyme is a biological molecule. Usually it's pretty small, meaning smaller than the enzyme. Or the helper could be a cofactor. Most cofactors are elements like iron, zinc, magnesium, calcium. One coenzyme is NAD plus, and we'll talk about this today, actually, this coenzyme. You do not need to know the names of the enzyme without the helper or the enzyme with the helper, but you can see here the coenzyme actually fits into the active site of the, well, not the active site, fits into the enzyme to make the active site then bind to the substrate. So it helps the enzyme form the active site, so the active site binds to the substrate. Any question about any of that? The helper can also help the enzyme take on its final three-dimensional shape, which can help, once again, the enzyme bind to the substrate. There are some important coenzymes, and we will discuss the first two in this lesson. There's NAD+, also called nicotinamide adenine dinucleotide, and that name is probably no more familiar to you than NAD+, so I would just call it NAD+. It's a niacin derivative involved in catabolic reactions, and it's involved in aerobic respiration. That's why we'll talk about it today. FAD is another important enzyme, flavine adenine dinucleotide. It also is involved in aerobic respiration, so we'll talk about FAD today. But beyond that, these are the only two coenzymes you must know. All the others you do not need to know, like NADP+. This is involved in photosynthesis. We're not going to talk about it. Oops, didn't mean to do that. FMN, I don't even know where that's involved in. It might be involved in aerobic respiration, but we're not going to talk about it. And coenzyme A, actually, we will talk about it, but I'm not requiring you to know it. Coenzyme A is involved in aerobic respiration. It's involved in, is that the preparatory step it's involved in? Yeah, it's the preparatory step. Anyways, no NAD+ and FAD, important coenzymes. They're involved in aerobic respiration, which is why I want you to know those two. And we will talk about aerobic respiration in great detail. Enzymes work by having their active site bind to the substrate, and the active site has a specific shape which only binds to one substrate. The two then form the enzyme substrate complex, which, let me blow this up. There we go. Which puts stress on the substrate, converting the substrate or helping convert the substrate to the products. And then the product can fall out of the enzyme. And now this enzyme is ready to bind again to another substrate. And that's what we mean with the starting enzyme is equal to the ending enzyme. The enzyme is not changed by the chemical reaction, at least changed permanently. It is changed here because it's forming a complex with the substrate. This is showing you the lock and key model of an enzyme, where a lock has a specific key to unlock it, and this enzyme can fit specifically to one substrate, and that's what we mean by the lock and key model. The lock and key model is a little oversimplified, and so they've come up with the hand in the glove model shown here. And what do you notice about this slide, or this part of the slide showing you the hand in the glove model? What can you notice? There's three things that an observant person can notice. What do you observe there? Come on, I'm not going to add this one, so you're not going to get it unless you...
Are you just talking about comparing the two in the middle slide?
Yeah, comparing this, that, and then this together. This is the complex. So this is actually chemically bound together as a complex. Okay, what do you notice about the difference here?
It's not as open for the substrate. I don't know.
The enzyme changes its shape. I think that's what you're trying to get at. The enzyme here, come on, mouse, here, it's changed here. And that's why we're saying this is the hand in the glove model. The glove molds its shape to fit the hand. Or in this case, the enzyme molds its shape to fit the substrate. So that's the first thing. Something's very obvious. What do you notice about the size of the different molecules?
One's a lot smaller than the other.
The substrate is much smaller than the enzyme. And that's usually the way enzymes are. They're very big molecules, and the substrates are usually very small molecules. The third one, you need a really observant student to observe this. But if you look very closely, let me blow this up, at the substrate here and the substrate there, you can notice a slight difference. This substrate is a little flatter than this substrate, or this one's a little bit more, I don't know, curvier. It has a little bit more of a up and down shape than this one. And that is what I like to call the shapeshifter's hand in the glove model. And that is, as the glove is molding to fit the shape of the hand, the shapeshifter's hand is molding to fit the shape of the glove. Those of you who have watched Deep Space Nine, you know what I'm talking about when I'm talking about shapeshifters, right? Odo was a shapeshifter and he could change the shape of his hand to make it fit the glove. Anyways, that's the way enzymes really work. Any question about any of that? All right, let's move on. Enzymes do not work in a vacuum. They do require a specific temperature, pH, salinity, that's salt concentration. They require usually the absence of heavy metals, the absence of alcohol, and UV radiation, the absence of UV radiation. If these are not correct, like you're at the wrong temperature, heavy metals are present, alcohol is present, UV radiation is present, the enzyme will not work at its optimal level. In fact, if there's lots of alcohol around or lots of heavy metals around, the temperature is really wrong, the pH is really wrong, even the salinity is really wrong. The protein of the enzyme can be denatured, and then the enzyme will not function. If it's totally denatured, the enzyme will not function. Any question about any of that? So these are some factors that can influence the enzyme's activity: temperature, pH, salinity, heavy metals, alcohol, and UV radiation. The substrate concentration can also influence an enzyme's activity. If the substrate is very low and you increase the substrate concentration, you'll increase the chance that a substrate will bind to an enzyme. So that will speed up the chemical reaction. There's a point to this, and that is, as you increase the substrate concentration more and more, eventually you will not increase the enzyme's activity any further. And that's because once all the enzymes are bound to a substrate, if you increase the substrate concentration further, you cannot speed up the chemical reaction. And we'll talk a little bit more about that in a little bit, hopefully today. Enzymes can also be influenced by the presence of poisons. cyanide and fluoride can destroy the enzyme. As you know, cyanide and fluoride are two toxins. I think everyone's heard of cyanide and cyanide poisoning. The same with fluoride. Too much fluoride is toxic. Generally, we do need a little bit of fluoride to help us form our enamel, but too much would be toxic. And I'm not sure how fluoride destroys enzymes, but it probably binds to the enzyme and then changes its shape. Enzymes can also be influenced by inhibition. You can have competitive inhibition, and you can have non-competitive inhibition. We'll talk a little bit more about both of these in a little bit. Non-competitive inhibition is also called allosteric inhibition. And then lastly, you can have feedback inhibition, which can also influence the amount of activity an enzyme has. Any question about any of this? All right, so an enzyme can lose its activity by becoming denatured. Here is the active functional protein, and as you can see, it is folded in a three-dimensional shape. And this would be a protein which is unfolded. It's no longer in this three-dimensional shape, it's in another shape. And this we call a totally denatured protein. And usually they just call it a denatured protein. There's different ways you can denature a enzyme. The simplest ways are put it at the wrong temperature, the wrong pH, or the wrong salinity. But you could denature it by adding heavy metals, alcohol, and UV radiation also. When we're talking about the temperature, all enzymes have an optimal temperature where the enzyme will work best. This enzyme works best around 37 degrees, which is our body temperature. So it's pretty safe to say that this enzyme is probably one of our enzymes. It'll work best at 37 degrees. When you go away from 37 degrees, a little bit less than 35, or a little more than 40, or actually just even 40 degrees. Come on. Sorry, my mouse died. The enzyme's activity will start to drop. Why? Because the enzyme is at the incorrect temperature and its folding pattern will change. It still has a fair amount of activity in this range here, but it's definitely less than this range here. And if you go further off on the temperature, the enzyme activity level will continue to decrease until you come at an extreme temperature. On this one, it looks like it's around 10 degrees Celsius, and this one, 50 degrees Celsius, where the enzyme will be totally denatured, and you'll see no activity from the enzyme. Any question about any of that? And that's about right. Our enzymes, you put them in the refrigerator, they're not going to work. 10 degrees Celsius, about 4 degrees to 10 degrees Celsius is, I think, refrigeration temperature. Here's an enzyme where we change the pH. All enzymes have an optimal pH. This enzyme has an optimal pH around pH 5. Where would we find a human enzyme that'll work best around pH 5? Come on, some of you have had A and P, I'm sure, so you should know this. Well, it's not the stomach because that's pH 2 to pH 1, pH 1 to pH 2. So this is not an enzyme that works best in the stomach. Nobody knows where you have pH 5 on a person?
Yeah, I do not know.
Okay. Your skin. Maybe your mouth also, but definitely your skin is around pH 5. So my guess is this is a human enzyme of the skin because its optimal pH is around pH 5. Come on, mouse. When you change the pH a little less than 4 or a little less than 6, the enzyme's activity is going to decrease. And as you change the pH further, the amount of the enzyme is going to decrease further until you get an extreme pH, pH 10, or in this case, pH 0. And then the enzyme will be totally denatured. The enzyme will have no activity. Remember that this isn't a normal human enzyme. That's an enzyme of the skin, because normally pH 0 is really, really acidic. That would be probably a little more acidic than your car battery acid. That's the most acidic thing I know of. And this enzyme has a little bit of activity, almost to 0, pH 0. You can't get less than pH 0. All right. This is just showing you that enzymes don't work in a vacuum. They have to have an optimal pH and optimal temperature. And when you move away from the optimum, the amount of the enzyme will decrease until the enzyme is totally denatured and the enzyme will have no activity. The substrate concentration can also affect the amount of enzyme activity. When the substrate is very low, if you increase the substrate concentration, you greatly increase the amount of enzyme activity. That's because the enzyme is much more likely to find and bind to the substrate if you start increasing the substrate concentration. But as you notice, once we get the substrate concentration above a certain level, the amount of enzyme activity begins to decrease. And eventually, right about here, when we increase the substrate concentration, it does not increase the amount of enzyme activity at all. And that's because about right here, all of the enzyme is already bound to a substrate. So if you increase the substrate further, you cannot increase the reaction rate at all because all of the enzyme is already bound to a substrate. Any question about any of that? All right, let's talk about another thing that can influence enzyme activity, and that is inhibition. You can have an inhibitor. This is a molecule which inhibits the enzyme. There can be competitive inhibition, and this inhibitor binds to the active site of the enzyme. Oops, sorry, right there's the inhibitor. Binding to the active site of the enzyme. When the inhibitor is there, that means the substrate cannot bind to the enzyme. So this is why it's competitive inhibition. The two are competing with each other for the active site of the enzyme. We do have some drugs which are competitive inhibitors. Any question about any of that? All right. An enzyme can also be inhibited by non-competitive inhibition, and this is also called allosteric inhibition. So the substrate binds to the active site here. The inhibitor does not bind to the enzyme in the active site. It binds in another site, and we call that the allosteric site. The allosteric site, when the non-competitive inhibitor binds to it, that changes the folding pattern of the enzyme, from this shape to that shape, and that changes the shape of the active site. Now this active site can no longer bind to the substrate. And that is how non-competitive inhibition, also called allosteric inhibition, occurs. The inhibitor binds at the allosteric site, and that prevents the substrate from binding to the inhibitor. Any question about any of that? All right, let's look at feedback inhibition. Feedback inhibition is a enzymatic pathway where you have, let me blow this up, one substrate which binds to enzyme 1 to make intermediate A, and then we have an enzyme 2 which takes intermediate A and makes intermediate B. And then we have enzyme 3 which takes intermediate B to make the end product of this chemical reaction. In feedback inhibition, the end product binds to the first enzyme, enzyme 1, in non-competitive inhibition, shutting down enzyme 1. Once you shut down enzyme 1, you do not make intermediate A. Without having intermediate A around, you don't make intermediate B. And with no intermediate B around, you do not make more end product. So that shuts down the chemical reaction. Any question about that part? Well, this is called feedback inhibition because the end product itself shuts down an enzyme which makes the end product. And this is one way that cells regulate the amount of products in their cells. They'll have the end product itself shut down the enzyme. So when the cell has enough end product around, it'll shut down all the enzymes and no more end product will be made. However, let's say the end product gets used up by some means, and so there will be less end product around. Now, if ever we have an enzyme 1 that is not bound to the end product, there will not be enough end product around to bind to it and shut down enzyme 1. Enzyme 1 will then make intermediate A, which will make intermediate B, which will make more end product. Any question about that? All right, so usually the end product binds in a stoichiomic reaction to the first enzyme. So every once in a while, this end product bound to the enzyme, the end product can fall off. But if there's lots of end product around, another end product will bind to it and shut down this chemical reaction. However, if there isn't enough end product around, then if this falls off, there will be no end product to bind to it, and then that enzyme will be active. It will make more end product. The cell will continue to make more end product until there's enough end product around to shut down enzyme 1. And then when it's totally shut down, no more end product will be made. And this serves as a check to keep the concentration of the enzyme within a certain range. At the high point, the end product will shut down the enzyme. No more end product will be made. At the low point, there won't be quite enough end product around. So when one end product falls off an enzyme, this enzyme will make more end product. Any question about feedback inhibition? All right, feedback inhibition is something seen in cells quite frequently. because it's one simple way for the cell to control the amount of this one end product, the concentration or range of this end product. The end product normally binds to the first enzyme, because if it were to bind to the second enzyme, you do shut down the production of the end product, but the substrate will be converted into intermediate A, and that costs the cell energy. The cell wants to be as efficient as it can, so usually the end product shuts down the first enzyme, because that's the most efficient way to engage in feedback inhibition. Any question about any of that? All right, not hearing anything, so let's move on. Let's talk a little bit about RNA enzymes. I mentioned that most enzymes are proteins. However, there are some enzymes which are not proteins. These are called ribozymes because they are RNA enzymes. They are not made up of proteins, they are made of RNA. The RNA that cuts and splices RNA or another molecule is an RNA enzyme. These RNA enzymes function as enzymes in that they have an the RNA enzymes have an active site that actually binds to the substrate. The RNA enzymes are also not used up or modified in a chemical reaction, and then they bind to their substrate as a protein enzyme does. So tutorial does not mention it, but I'm going to mention it. We will later talk about RNA and ribosomes that help make the growing polypeptide, meaning the ribosomes make the proteins. As the protein is being made, we call it a polypeptide, and the RNA portion of the ribosome actually links the amino acid chain to the new amino acid that was just added. And that is the only RNA enzyme we will talk about in this class. So that's an example of an RNA enzyme. It's part of the ribosome. It's an RNA part of the ribosome, and it links the growing polypeptide to the new amino acid that has just been added. Any question about that? So most enzymes are proteins, but there are a few which are ribozymes or RNA enzymes. Any question about that? If not, we're going to change focus a little bit and talk about metabolic diversity among organisms. All organisms, including microbes, require the following. They require an energy source and they require a carbon source. Based on their energy source, we can classify organisms as a phototroph or a chemotroph. Phototrophs derive their energy from light. Chemotrophs derive their energy from a chemical compound. And that chemical compound could be an inorganic molecule like hydrogen sulfide or sulfate or nitrate, an inorganic molecule, or from an organic compound such as glucose. If you don't know, humans are chemotrophs. We get our energy from an organic compound such as glucose. You can also classify all organisms depending on where they get their carbon source. An autotroph is an organism which gets its carbon from CO2, either in the air or dissolved in the water. A heterotroph gets its carbon source from an organic chemical molecule containing carbon. We are heterotrophs because we get our carbon source from glucose and any other food that we eat that has carbon in it. Any question about any of that? So remember, if you get confused on the different classification of organisms, that you can just break it down. What's their energy source? If it's light, they're a phototroph. If it's not light and it's a chemical, then they're a chemotroph. And what's their carbon source? If they get their carbon from CO2, they're an autotroph. By the way, green plants are autotrophs. And if they get their carbon from an organic chemical like glucose, they are a heterotroph. And humans are heterotrophs. Now you can combine these two by classifying the organism on where they get their energy source and their carbon source at the same time. These make the classifications of these four a defined classification for all organisms. There's a photoautotroph, and these are, well, I'll talk about it later. There are photoautotrophs, there are photoheterotrophs, there are chemoautotrophs, and there are chemoheterotrophs. Let's go to the next slide and I'll talk about each of these. A photoautotroph is fairly easy because green plants are a photoautotroph. Green plants get their energy needs from light and they get their carbon from CO2. So green plants are a photoautotroph, so are green bacteria like cyanobacteria, which actually they're blue-green in color, but they're photosynthetic. That's what I mean by green microbe. There's oxygenic photoautotrophs, and they make oxygen, and there's anoxygenic photoautotrophs, and they do not make oxygen. And we're not going to talk about the bacteria that are anoxygenic, but green plants and cyanobacteria are actually oxygenic photoautotrophs. I'm going to skip photoheterotrophs because I'm sure you've never heard of them, or at least the species. We're going to skip chemoautotrophs just for a little bit and then talk about chemoheterotrophs. These should be familiar to you because humans are a chemoheterotroph. A chemoheterotroph gets their carbon source from an organic chemical compound such as glucose. And humans get their carbon from an organic compound such as glucose, which we eat, or sucrose or other carbohydrate, which can be broken down into glucose. But any food source you have will contain, well, almost any food source you have will contain carbon, and then you can burn it to get the carbon source. Candy would be an exception. And I suppose if you were to drink oil or eat butter, wait a minute, butter? I take that back. Butter has carbon in it, so does oil. So the only thing I can think of which we sometimes eat that doesn't have any carbon in it would be candy. There might be something else, but most things we eat do have carbon in it. Okay. And we get our energy source from glucose or another food we eat. And that's why we are a chemoheterotroph. Our energy source is a chemical compound. Our carbon source is an organic chemical compound. you should know that chemoheterotrophs have a chemical energy source, but it can be or an organic chemical like glucose, or it can be an inorganic energy source like sulfate or hydrogen sulfide gas. And we'll talk a little bit more about that later. Obviously, we can only get our energy from an organic chemical compound such as glucose. It's the some of the chemosynthetic bacteria which derive their energy needs from an inorganic chemical compound such as nitrate or hydrogen sulfide gas or sulfate. So some of the chemosynthetic bacteria are chemoheterotrophs, and they get their chemical from an inorganic chemical source, their energy source from a chemical, sorry. All right, let's talk about the chemoautotrophs. They get their energy source from a chemical, inorganic chemical compound like hydrogen sulfide gas, sulfate, nitrate, and they get their carbon source from CO2. Why they are a chemoautotroph. The only organisms that are chemoautotrophs are the chemosynthetic bacteria and the chemosynthetic archaea. And you'll note that some of the chemosynthetic prokaryotes, meaning bacteria and archaea, are chemoautotrophs, and some of the chemosynthetic bacteria and archaea are chemoheterotrophs. Any question about that? The difference is where they get their carbon source from. If they can get it from CO2, they are a chemoautotroph. The chemosynthetic bacteria who get their carbon from an organic compound such as glucose are a chemoheterotroph. Any question about that? All right. Are you, as I told you, you know a chemoheterotroph because humans are chemoheterotrophs. You do know a chemoautotroph. You just maybe don't know it yet. But I'll bet all of you have lifted up the toilet tank that stores the water to flush the toilet. And you see some yellowish brown or brownish scum on that toilet tank, either the bottom or the sides of the toilet tank. That is actually iron oxidizing bacteria, bacteria that get their energy from iron dissolved in the water. And of course, they get their carbon from CO2. So all of you are familiar with the chemoautotroph because I know all of you have seen that brownish or yellowish scum on the toilet tank. Any question about any of that? All right, the photoheterotrophs get their energy source from light, and they get their carbon source from an organic chemical compound such as glucose. An example of a photoheterotroph are green and purple non-sulfur bacteria. And I'm sure all of you have heard of green and purple non-sulfur bacteria, so I don't need to discuss it further, now do I?
You can if you want.
Yeah, I'm not going to discuss it further. I know none of you have you're familiar with green and purple non-sulfur bacteria, except for this slide. I've never come across green and purple non-sulfur bacteria in all my years of teaching microbiology. So I'm not terribly familiar with them. They are a bacteria and they're a photoheterotroph getting their energy needs from light, but they get their carbon from an organic chemical compound such as glucose. All right, any question about any of this? Oftentimes with the chemoheterotroph, the same molecule is used for the energy source and the carbon source. So like for humans, glucose is our main carbon source. It's also our main energy source. That isn't always the case, but it is often the case. Let's go on to the next slide. You must know the groups surrounded in the red box. We can separate all organisms by their energy source. If they get their energy from light, they're a phototroph. If they get their energy from a chemical, they're a chemotroph. We can then split the phototrophs by where they get their carbon source. If they get their carbon from CO2, they're a photoautotroph. If they get their carbon from an organic chemical compound, such as glucose, they are a photoheterotroph. Likewise, we can split the chemotrophs, getting their energy source from a chemical compound, into those who get their carbon source from CO2, and those are the chemoautotrophs. And then we can also split those chemotrophs by those who have a carbon source, which is an organic chemical compound, and those are the chemoheterotrophs. Any question about any of that? You should know that. I'll briefly discuss that we can split the photoautotrophs further on whether they use water to reduce CO2 and produce oxygen. If they do, then they are oxygenic photosynthetic. All green plants, algae, cyanobacteria are oxygenic photosynthesizers, but you don't have to produce oxygen in photosynthesis. You can have anoxygenic photosynthesis, and they do not produce oxygen. We're not going to talk about the photoheterotrophs because we're not going to split them. The chemoautotrophs, I've already talked about them. You can split the chemoheterotrophs into those which have oxygen as their final electron acceptor, and these are all of those that engage in aerobic respiration. So that would be all animals, fungi, protozoa, and most bacteria, but not all bacteria. And you can split the other group into those who the final electron acceptor is not oxygen. They do not require oxygen because they don't use it. And those would be the anaerobes. All right, any question about any of that? If not, let's move on. Energy production. Nutrient molecules have energy associated with the electrons that form their bonds. In cells, energy is obtained from catabolic reactions and stored in the bonds of ATP. So really, we're only talking about catabolic organisms, and they obtain their energy from a catabolic reaction using the energy stored in the bonds of the molecule to make ATP, and then that ATP can be used whenever the cell has an energy need. ATP is an energy carrier of cells. By energy carrier, we mean ATP has a high energy unstable bond, and then the cell can break that bond to quickly release a lot of energy for the cell's energy needs. Any question about that? One energy need the cell needs is when it is making a larger molecule in biosynthesis. You need to add energy to get the biosynthesis reaction to occur to make the larger molecule. And the cell usually will break down ATP to get the energy to make the biosynthetic reaction occur. Any question about any of that? All right, let's go on to energy production in cells. Energy production occurs, at least in catabolic reactions, by a series of oxidation reduction reactions. And that's actually true of photosynthetic cells as well, that energy occurs by a series of oxidation reduction reactions. Oxidation is the loss of electrons. So this molecule loses an electron. We then say A is oxidized because it lost the electron. Reduction is the gain of an electron or electrons. So B gains the electron. And then we say B is reduced, the gaining of an electron. Oxidation reduction reactions are always coupled. One molecule will lose the electron, the other molecule will gain the electron. Because oxidation reductions is a fairly long word, people came up with a simpler term and they call it a redox reaction. A redox is simply an oxidation reduction reaction. So they took re from reduction, ox, or actually red from reduction, and ox from oxidation to make the redox reactions. Here's an actual example where we have copper +1 plus iron with 3 positive charges can engage in an oxidation reduction reaction to make copper 2+. So copper is oxidized because it's losing an electron, +1 to +2, meaning they lost an electron. And then iron gains the electron, so it is reduced. It was +3, and now it's +2 because it's gained an electron. If you don't remember, an electron has a negative charge. So how do you remember oxidation and reduction reactions? Specifically, that oxidation is a loss of electrons, reduction is a gain of electrons. Well, here's a little mnemonic for you to remember that. Leo, the lion, goes grr. Leo, lose electrons, oxidation. The lion goes grr, gain electrons, reduction. Any question about any of that? All right. In biological systems, oxidation usually has a hydrogen ion being removed with an electron. Remember that hydrogen ion plus the electron equals the hydrogen atom. So in biological oxidations, we often call them dehydrogenations because the hydrogen atom is also lost along with the electron. And the same with reduction in biological systems. The proton, the hydrogen ion, are typically gained when the electron is gained. An example of that is the organic molecule, like glucose, loses an electron, which is picked up by NAD+. The organic molecule, such as glucose, loses a hydrogen, which in this case is also picked up by NAD+. And then our organic molecule is oxidized because it lost an electron. And NAD+ is reduced because it gains an electron. In addition, it gains a hydrogen. And that's just what happens in biological systems, where the hydrogen often follows the electron. Any question there? All right, if there's no questions there, I think I'm gonna stop here. Well, yeah, I'll stop there. And I'll see you at 6:30 in the lab unless there's a question. All right, I'll see you in the lab.